Hey guys, welcome back. We're still in Chapter 8, Genetics of Behavior, and this time we are moving on to Section 3, which is Intelligence and Intellectual Ability, as well as Section 4, Drug Addiction. So I hope you guys are ready for this. So let's get going. So intelligence is really ill-defined. If I was to ask you, or to ask anyone, what is intelligence? How would you define it? Most people would say how smart you are. Okay, well, what does that mean? How do you define how smart you are then? And the reason why it's so ill-defined is because it's a complex trait. And there are quite a few genetic components as well as quite a few environmental factors that come into play. Now, people thought they had it figured out back in the early 1900s when they came up with IQ tests. That's when they were first developed. And they looked at four main things: verbal fluency, math reasoning, memory, and spatial visualization. And the thing is, while these scores were useful predictors of success in school, they were by no means an end-all be-all to someone's intelligence, meaning that there's a lot of other factors that go into it besides just that one score. I mean, they could predict if you could do well because it looked at these different factors, but there's a whole bunch of environmental factors it didn't take into account at all. So that's why we had to be very careful in labeling individuals or labeling groups of individuals based on those IQ scores. Because once we label them that way, they tend to stay that way, and society kind of has a stigma with certain labels. Now, what I mean by this is, imagine that you are very highly intelligent. However you define intelligent, you are very highly intelligent. Now, let's say you broke up with your significant other and you're someone you care very deeply about, whether it's a parent, a sibling, they just got diagnosed with cancer, and your beloved dog dies. No matter how smart you are, those factors are going to influence your score, and they might end up listing you as a very low IQ, which now everyone thinks means that you're dumb, stupid, maybe that you have some kind of mental impairment because you scored so low on it, and people are going to treat you differently because of that. But in reality, it wasn't because you weren't intelligent. It was just because there's all these other factors that can affect your ability to, maybe remember certain facts, or if you're too busy stressing about personal things, you may not be able to fully work out the reasoning behind a math problem or see the pattern for spatial visualization. So this is something that we have to be very careful about. Now, there's different IQ tests out there. I do have some sample questions here though, if you are interested. So essentially what they are is find the pattern. What would come next? So for this one, it's essentially asking you, what is the pattern? How do you figure out the pattern? For this one, it gives you some of the questions. I'll move this over. All right, if this means equals that, this equals that, this equals that, then what would be the answer here? Some of them will give you answer choices. All right, so if you have little star, medium star, big star, little, that shape, medium, big, little, medium, what would go here? I'm hoping you guys are saying this one. Now that might seem kind of easy. It doesn't mean they all will be. We have ones like this, boxes, circles, and X's. We have this one. All right, what goes next? So there's a lot of different things going on. Or here. So again, lots of different things going on where they want you to figure out the math, the reasoning, the visualization, the comprehension. That's actually what goes into a lot of these IQ tests. They're not necessarily facts. They're looking at patterns and or mathematical problems and trying to come up with solutions. There we go. Let's talk a little bit about the genetics of intelligence. Well, genetic disorders that affect neurological function can affect intelligence in patients. Single genes that affect intelligence are subject of an intensive search. A few candidate genes have been identified. So what we're saying is that there are things that we are looking at in the brain, in neurological function, that could affect someone's ability to learn or retain information. There are genes associated with building more connections between neurons as we learn different things. So as you read more, as you learn more, you do tend to pick up more neural connections. Well, you have to be able to build those. What if something in your genetics doesn't allow you to build those as quickly? What if, let's say, these stimuli from your eyes, the visual stimuli from your eyes to your brain, there's a misconnection in that neurological pathway and you don't see things the way other people do, like with dyslexia. Someone with dyslexia will see the words kind of moving around, letters moving around. They won't see the words as clearly as someone without dyslexia. You have two people looking at the exact same sheet of paper, but they can actually perceive it differently. And it's because the signal is getting misinterpreted somewhere between the eye and the optic nerve and the brain. So these are all possible candidate genes to look at when we deal with intelligent or genetics of intelligence. And there's actually a lot more in that. I don't have time. Any one of these subtopics for behavioral genetics, like intelligence, mood disorders, addiction, sleep disorders, eating disorders, any one of those we could spend an entire year looking at. So right now we're just trying to summarize everything. All right, let's talk a little bit about, let's actually go back to intelligence for a moment. Let's talk a little bit about the environmental factors that can go into that. What I mean by that is, even if you have all of the best genes, so you have the alleles that promote neural network growth and neural connections, you have genes that allow for signals to be interpreted properly and quickly, but you may not be perceived as intelligent as someone else depending on certain environmental conditions, such as there is no denying that children in lower socioeconomic conditions are at a disadvantage in terms of not supplies, resources, sorry. I'm a little tired, I apologize. But in terms of resources, they may not be able to have good access to books. Maybe they don't have access to consistent internet or technology. And maybe they have to babysit their younger siblings as opposed to studying. So even though they have these genes that would suggest that they are highly intelligent, because of these environmental factors, they're not able to reach their full potential. As opposed to some other people who maybe have poorer genes, I shouldn't say poorer genes, but genes that wouldn't necessarily facilitate large neural connections or neural networks, or that sometimes garble up the signals going to the brain and misinterprets them. But if they have a lot of access to resources, state-of-the-art private schools, fast internet, incredible technology, private tutors, being able to travel to different places around the country and around the world to see where historical events, or to see museums and things of that nature. I mean, then they are even though genetically they're at a disadvantage because of their resources and i.e. the environmental factors, they can be perceived as more intelligent. So there are these environmental factors to take into consideration. All right, let's look at drug addiction. So drug addiction is a compulsive behavior that exhibits two main characteristics: tolerance and dependence. Now, tolerance is when you no longer get the same effect from a stimuli, in this case drugs, and so you have to take more of it to reach the same effect. Dependence is just when you feel like you need it, where if you don't have it for a while, you end up going through withdrawal symptoms because your body is freaking out that it no longer has this chemical or this compound inside of it. Now, they have done imaging techniques, and they've used these to identify the different regions of the brain that are involved in drug addiction. For instance, at the bottom here, this is someone who is not addicted to anything. So then this is someone who has been on cocaine after 1 month. You can see the colors indicate dopamine D2 receptors. So you can see how that's different from there. And after 4 months, you can see some slight differences again. So what this is showing is, as it says down here, low dopamine D2 receptors may contribute to the loss of control in cocaine users. So as someone becomes addicted to cocaine, their dopamine D2 receptors decrease. Now, dopamine is one of those happy hormones. It helps us feel good. And essentially what happens in someone who is addicted, our bodies naturally produce dopamine, as well as the receptors for it. Now, when we start taking a drug, the drug is hitting those receptors, which means that we are getting the effects, but without our bodies producing the dopamine itself. The drug is taking over for it. So our bodies will actually stop producing some of those different hormones or the receptors for them because the drug is taking care of all that for us. As a result, that's why withdrawal is so bad, because let's say here, the dopamine wasn't being produced by the brain because the cocaine was causing the effect that dopamine usually would. And that's, euphoria, the happiness, I've never been on cocaine, so I'm only going off what I see and what I've read. However, when they are no longer addicted, or when they are going through withdrawal, they no longer have anything to stimulate those receptors. So now they don't even have their own natural dopamine because their brain stopped making as much because the drug was taking care of that job for it. So now that's why it gets really low, and why they start having all these different people who are going through withdrawal can become very depressed because of these low levels. There's also a lot of physiological effects, such as it can affect different organ systems, it can affect metabolism, it can affect how the brain just reacts to different things, not just dopamine, but different things in general. All right, and over here, Again, showing controls, or what they mean by controls, people who are not addicts. And then this is showing, again, receptor availability based on if they're addicted to cocaine, meth, alcohol, or heroin. In all of them, you can see it's definitely different than the controls. All right, let's move on. So genes implicated in drug addiction are involved in biosynthesis of neurotransmitters, reuptake transporters, neurotransmitter receptors, and components of signal transduction pathways. So pretty much what that means is we are looking at the genes that make the different hormones such as dopamine, as well as the receptors that both the dopamine and the drugs have access to. And what they found is that there actually is a genetic component to addiction. You might have heard people say how alcoholism runs in families. Well, that's because there are certain genes related to these transporters and receptors, and just in general, the neurological pathways within the brain. There are these different genes that make it more likely, because they affect these systems in the brain, but they make it more likely that someone could become an addict. Now, again, it means you're predisposed. It doesn't mean you will be. Essentially, what it means is someone who is predisposed to be an addict, it would require less of the stimulus for them to become addicted. So let's use the example of alcoholism. Maybe it would take an average person 4 weeks of intense drinking before they become addicted to alcohol. Now, I'm not saying that is the case. I'm just using it as an example. Someone who is predisposed, who has a genetic predisposition to alcoholism, it might only take them 2 weeks of intense drinking, because after that point, they were more susceptible to the alcohol affecting the signal transduction pathways, and it only needed a certain amount, a lesser amount of the alcohol before the dependency kicked in. However, like we said, there are environmental factors. If I never once choose to start drinking, if I never once choose to drink alcohol, but I have a genetic predisposition to alcoholism, but I never once drink, I'm not going to be an alcoholic. So again, a lot of these environmental factors are very, very strong. Also could be, things like peer pressure. And I'm not just saying that from, other kids, but even adults can peer pressure each other into drinking frequently. Or a lot of times people might use drugs as an escape because something in their life just, maybe it's their home life, maybe it's they're in an abusive relationship, whatever it is. They just want some kind of escape. They want something to make them feel good for once. Let's see. DNA microarray tests are revolutionizing the analysis of addictive behavior and demonstrating the pattern of gene expression during drug use. So what this means is we're able to use these particular tests called DNA microarrays, and they allow us to actually look to see which genes might be involved and which versions of them are being expressed more frequently in addicts versus non-addicts. Let's look at some examples. So certain drugs are able to affect us because they bind to receptors on human neurons. Now, what we mean by that, readjusting, is you've heard me talk about receptors on our cells and all that. Now, if you remember from biology, receptors are extremely specific. It's not like just anything can attach to them and cause a reaction. But the reason why certain drugs can so easily bind to our receptors is because we actually have similar versions of these chemicals that we make already. We mentioned dopamine, but there are other certain chemicals found in plants that are very similar in structure to the ones we make. Using dopamine as an example, we said dopamine is a happy hormone. It makes us feel calm, content, happy, things like that. Now, since we can get, let's say, from heroin, it can attach to those receptors and activate them. But since it is not the exact structure, it's close enough in structure that it can attach to the receptors, but it's not the exact same structure as dopamine. That's why we see such a different effect. Dopamine makes you happy. Heroin might make you feel, like you're in a state of euphoria. So we do see these stronger, more intense effects from the drugs because they're not exactly the same structure as chemicals we make. They're close enough that they can trick the receptors, but different enough that they still have this stronger effect. And a lot of these tend to be plant-based, so like cocaine, opium, THC, etc. Here's just kind of a picture here. So we have dopamine being transported. We have the receptors, and then we also have the dopamine transporter being blocked. What that means is a lot of times once the neurotransmitters are released, they will actually be reabsorbed back here. But what happens is the cocaine fits into the receptors, and that prevents the reuptake of the dopamine. So you just have this intense flood of dopamine that just keeps hitting the receptors over and over and over and causes an overstimulus of them, hence the high you feel. And it's just kind of showing it closer up. All righty. Well, that's it for sections 3 and 4. I hope this helped you guys to learn a little bit more and understand a bit more. Like I said, I know we're not going into too much detail. We can have an entire year's course on any one of these topics. But I hope that you are still finding it informative and interesting. If you have questions, hit me up in our Google Classroom or through email. Otherwise, I will see you guys next time. All right, guys, take care. Stay awesome. Stay curious. Most importantly, stay safe and healthy. All right, you guys, take care. Bye-bye.